So now I'm actually in Sweden at SciLifeLab. So actually, when I talk with my colleagues, and then my American colleagues told me that, hey, Wei, you have a cute, French accent. So that was super interesting, and also kind of I kind of enjoy a lot my time, in France. Actually, it's good to be with the community again. So I'm currently in this Swedish National Center SciLifeLab. It's a research environment. We have a lot of infrastructure in the same building with for life sciences. And I was joining this data-driven life science program as a kind of DDLS fellow. So it's a kind of a dedicated program for recruiting a lot of young PIs and to push the boundary of next generation life science, which is more AI and data driven. So more recently, we have this, actually Jan Ellenberg was joining us as a director at SciLifeLab. So he's gonna started this new program called Alpha Cell Initiative together with Matthias Uhlen, who is this director of the Human Protein Atlas. So it's a lot of exciting opportunities ahead with imaging and also AI, to be in the SciLifeLab. So today I'm gonna share a bit my kind of initial. Actually, the lab is quite new. We are focusing on this generative AI for image and modeling. You have been maybe seen a lot of DALL-E and this kind of text-to-image AI models, and also video generation, protein generation. There's also large language models everyone is using now. There's also, on top of that, you build AI agent, which is a great tool for, I think it's a great potential in controlling microscopes. So Gen AI in general enables, for example, complex system modeling, including human cells. This is what we are interested in our lab. And also, it has a great potential in automate the data analysis in instrumentation, namely the microscopes, and all the way to this automated scientific discovery. So all the work I'm showing here is quite of kind of work in progress, but I'm super excited to share. So my lab is called AI Cell Lab. So the aim of this lab is trying to build a human cell simulator. It's obviously an ambitious goal. But it has to start from somewhere, right? So our focus is mainly on this life cell imaging and combined with generative AI, trying to simulate the cell in a black box manner, which is less ideal, but I think it's just a different way of tackling this modeling problem. And the idea is to enable in silico cell experimentation, drug screening. Maybe later we can understand how it works, but this is a kind of secondary priority for us. The approach is quite straightforward. So we basically take the real cell, put it on the real microscope, and then you generate images and videos. And normally you just run some image analysis algorithm to extract phenotypes. So what we do instead, we build an AI model to represent the human cell, the real one, and it's a virtual cell. And then we also add another AI model to represent the real microscope. It's a virtual microscope, and then you connect the two and ask the whole system to produce synthetic images, just like what Flavius was showing with these actin images, for example. So the goal is that you generate this arrow, and then you do back propagation to run through these two models, and then you train it end to end. So in practice, this we could, simplify it as a kind of video diffusion model. So basically, you're trying to use this AI model to produce videos which looks like real. So this is what we have been trying so far. It's quite preliminary, but I'm still happy to share with you. What we tried with this Cog Video X is a diffusion model. It's a diffusion transformer. It's close to this OpenAI Sora. If you see a lot of videos about that for natural image or movie generation. So what our data set have been trying is this Newman is from this very old publication, actually from Jan Ellenberg lab. So and then this is it's a kind of siRNA knockout data set. So they did a lot of screens, and this is how the cell looks like. And then what we tried is take this off-the-shelf Cog Video X model, and then we ask it to generate this, video of cells, and this is the prompt that we fit in. So if you look at them, it actually looks quite artistic, isn't it? So obviously it doesn't work because it has no training. So what we did instead is trying to have this training. So basically we take these videos, feed into these models, and then we also give text input. The text prompt example looks like this. And then we have an initial frame. So we feed the first frame to this model, and then there's some kind of phenotype embedding. This is a kind of different combination we have been testing. I'm happy to share you with some kind of preliminary result. So on the left is real movies, and on the right, these are kind of the fake movies you can see. So the goal is obviously not just publish, fake videos, but the goal is to actually, if you see similar behaviors and which is undistinguishable from human eye, which is very high standard, compared to, existing kind of quantitative approach in many cases. So the goal is that we want to use these generated images as a supervision to the model. So we know this model captures this kind of, genotype to phenotype mapping. And this is kind of one of the conclusions of the goal, of kind of vision of the goal. We want to build a blurry JPEG of the human cell. So this is from the same ChatGPT blurry JPEG of the web. The idea is you generate a lot of observations from the microscopes, and then you're trying to train through this kind of generative AI model training the diffusion models, you're trying to compress as much as information as possible. And during this compression process, you're gonna naturally gain this kind of knowledge and concept, during the process. Yeah. So then you have this model, you can do sampling, you get this kind of synthetic images as a way to supervise the model. This is quite impressive so far with this generative AI development. For example, DeepSeek R1, for example, is only 715 gigabytes on disk. But imagine that you can run it on your laptop, and it has entire kind of human knowledge almost inside this laptop, which is super impressive, right? And then we want to do the same. We want to have a compressed understanding of the cell just in this diffusion model, for example. So this is kind of what we have been doing so far, and it has been quite difficult. Although you see this, seemingly looking nice images, but actually, in order to generate this kind of a conditional images is quite difficult. For example, if we give a protein sequence to the model, and then the model cannot generate the properly according to this condition. Our understanding is that it's mainly due to lack of data. So this is a kind of number comparison. The original Cog Video X has 35 million videos, but we only use 3,000 videos, right? So this is too far away. We need more data. And importantly, we also need the infrastructure to analyze the data. So this is the second part of my talk, which is one of the current major focus of the lab is trying to build the infrastructure for generative AI-powered bioimaging. So I'm going to talk about two parts, some kind of data platforms, and also this smart microscopy all the way to this AI scientist as a long-term vision. So one part of the goal of this when we start is that trying to move away from the desktop software, because this is the only way to scale, because you're going to handle much more data to move to the cloud. right? So this is one of the, actually, when I finished my PhD, I studied this project called ImJoy with Florian, who is also here. So it's quite, the ImJoy is a kind of web-based applications. Yeah, so we have one of our most popular applications, Imager JavaScript, where you can run Imager on your laptop or, the iPad, for example, or your mobile phone, which is quite neat if you don't want to install. And then after that, we have been working further to extend ImJoy to the cloud. So HiFi is this platform which extends the capability from the web browser to the cloud. HiFi is a kind of a open source library in Python. And then around Python, we build this HiFi app engine, which is a kind of Kubernetes cluster-based compute engine with a lot of other components. One of the examples is that HiFi has been used for support this bioimage model Zoo, which is one of these AI for Life consortium with many partners from the EU. We have been finishing in end of August this year for 3 years. The results are quite exciting. We have the standard, we have the model zoo, we have a lot of models from the community. But from our part, we have been building this bioengine, which is on top of HiFi. It's an infrastructure for serving AI models from the model zoo. So basically, it enables the user to upload images from the browser, and then you can run this without any installation. It's just a way to try these AI models before you kind of buy in a lot of, set up a lot of content environment, for example, is very complex. Yeah. So, and then HiFi has been extended also for other purposes. For example, enable human-in-the-loop image annotation. So we have this bioimaging collab, which is an open source tool. Is just go to this website, and then you can select your local folder with a lot of images, and then you can create this annotation session. Then you get the link like this, right? And then you share it with your other colleagues also, and then, hey, could you please annotate these images for me? And then the other ones, your friends or colleagues receive an URL, and then they can start annotating on their mobile phone or iPad, for example, or just in any laptops, and then they can, run this, AI-assisted annotation. So, for example, they can just click, like this, yeah. And then when they click save, it actually end up in your own laptop, in your own kind of local folder. This is kind of quite practical tool to crowdsource annotation, let's say. And then for the next part of my talk, I'm going to talk about AI agent for bioimaging. So we have been focusing on the other part of the, Gen AI, which is language model based AI agent, right? So what is an AI agent? An AI agent is a language model equipped with different tools. The tools can be any, let's say, Python functions. Let's load your data, snap an image from the microscope, or set the exposure, or this sort of tools, yeah? So you can connect these tools to a language model, and then you provide instructions as a prompt. And this makes an AI agent. So nowadays, it's a super powerful approach to build this kind of agent, multi-agent system. So with HiFi, is naturally designed for this type of purposes. So HiFi as a central hub allows to connect different AI agents together. So we have been working on this bioimage model, bioimage IO chatbot, which is a kind of a retrieval augmented generation system. So basically, we connect a lot of tools, such as some kind of documentation tools, online APIs, models into this language model, like OpenAI GPT models, and then it enables this kind of question answering with less hallucination because it's grounded on this community contributed bioimage analysis documentation, for example. And what's more, it also allows this, for example, you can mount your folder of images, and then it can generate code. Say you can do segment the images with cell pose, right? And then it generates the Python code and runs immediately, and then allows you don't need to know so much about code. You still need a bit of knowledge about how to do, image analysis in general, but this kind of makes it so quick, right? To explore different ideas and, and then get this kind of result quickly. So for example, in this case, it's just do this. It also fixes the errors. This is what we are showing. I'm gonna quickly go through this. So what this means, right? It means that we can provide a new way of doing interacting with bioimage analysis software, right? So because ImageJ, for example, is a great tool, classical tool, but also because of great contributions from the community, it's getting complex. Yeah, it's super difficult to use, it's super powerful, but the thing is that it's hard to use afterwards. So with this kind of like a chatbot, for example, it gives you this opportunity that you can simplify the interface and still make it super powerful. And what's more interesting is that when you equip this Python code interpreter to the language model as a tool, and this enables next level possibilities, what we call code agent. Code agent means connect a tool which can run arbitrary code. Of course, you need a sandbox to run this code in practice, but this allows you to access to hardware like GPUs, like microscopes. And this is a great combination. And then also we have, it's super popular nowadays, using this model context protocol to connect these tools to LLMs. It's MCP, right? So this is, you might heard it many times and many places, and everyone's building this MCP. So basically, if we can build a microscope, build an MCP interface for the microscope, you basically enable controlling your microscope from ChatGPT, for example. And then this is essentially what we are trying to enable with Haifa. So we're trying to enable this cursor, cloud code, Windsurf, and this different type of code agent to access the microscope and access the image analysis functionality. So this is one quick demo. It's not live, but the idea is that we have this malaria data set. It's a collaboration with Stanford Manu Prakash Group. So we have been building this portal where you can just type in, I want to create an app for segmenting the parasite images in the parasite ring in the image, and then trying to produce, have an analyze button on the interface, and then we would like to see the morphological distribution. So this is the only sentence you give to this portal, and then you click generate the app. So this is not real-time video, but the actual time takes 2 minutes. And in 2 minutes, it's literally one shot, and then you get this app created in this portal, and you click launch the app, and then you get this very nice web UI with real analysis functionalities. You can select a sample, then you click the button, say, I want to analyze this image, this sample. You get this very nice plot of distribution. And so because it's code generation, how can I know whether it works, right, without inspecting the code? So you kind of gain confidence when you see the interface. Because if you look at the rings of this is a red blood cells with a malaria parasite, and you can see that it's actually a segment, right? You can see the generated app is actually doing the right thing. So even if you don't know code, you kind of you're trying to have a way to inspect, whether the model is doing the right thing. So this is kind of actually make it super powerful. We can also use this AI agent for controlling our microscope. this is a kind of quick demos that you can, if you connect this microscope through this MCP interface, then you have this language model you can talk to through this, kind of, you can provide some tools, snap image, move the stage, and things like that, right? And this is actually enables a lot of other possibilities. I'm actually in time. So basically, you are stepping away from this very complex, very flexible, classical user interface to a chat interface, and all the way to this personalized GUI. Yeah? So with this code agent, it allows you to generate this customized, GUI. For example, I come to the lab and then say, I want the snap button into pink or something, right? It's gonna customize immediately for you, right? So this is, it's also a very interesting approach is that it's reproducible. If you work with the ChatGPT, for example, you know that it's not reproducible. You say the same thing the next day, it generates different result, right? So that's why you don't want to always interact with the kind of language model, because it's not reproducible and it's also not deterministic. With this code agent, the generated code can be saved and can be retrieved. It's actually made it reproducible and deterministic. And also it makes it much more kind of user-friendly. So this is kind of next generation of user software design. And it's in the future, you're gonna see a lot of UIs which generated dynamically. Imagine that when you open a software, there's nothing, but then you just started to interact with it. It generates this UI dynamically for you. So we have been also using these different technologies to put together to build this Reef Smart Microscopy Imaging Farm. It's a work with my PhD students and postdocs. And this is, the idea is to produce a lot of data to, for the whole cell modeling project. So this is what we have. We have the liquid handling robots. And then in the meantime, we have this automated imaging system where we have, the aim is to have like 10 microscopes on an optical table with robotic arm. This is kind of an incubator where you can go back and forth entirely kind of remote and controlled by AI. This is also a collaboration with this SciLifeLab company, which is a Stanford collaboration, a spin-off from Stanford. So they have this very nice, customizable, but still very simple, not as fancy as many of the microscope I see, during the workshop, for example. So this is an interface which also generated by AI. So with this kind of interface to the microscope, then you can, it enables a lot of possibilities. You can just use AI to, this is made by my student who don't know any kind of front-end programming, but he managed to generate this very rich and, microscope UI with this very nice 90-degree plate, and then they can click and then do that, right? So it's actually fundamentally changing the way how we build software. This is another example that you can see that if you embed this chatbot together with this UI, and you can do even more advanced stuff, right? You can type a sequence and then, I say, for example, it can help you generate configuration onto like a multi-dimensional acquisition, right? Which can be sometimes tricky to set up. I'm gonna check if I can actually do this live demo. I'm not sure the Wi-Fi will work. If not, I can go back to the videos, okay? Okay, so I'm actually trying to do this demo which connect to our Reef image farm in Sweden. Let's see if we are in luck or not. I'm gonna actually try with my mobile phone network because it's a bit slow here. Okay, yeah. If you're watching on Netflix or something, please stop. Okay, so what you're seeing is this agent lab, which is a kind of a notebook-like interface where we use to build the AI agent. So this is actually some kind of environment setup where we provide the necessary tools for the agent. And then I'm going to actually refresh this. So just to browse, actually, I'm going to make it bigger so you can see. Okay, so this is just some Python code, yeah? It's, we just define a lot of Python functions. This is what we used as the tools for the agent. And then we are running it. It's gonna produce some kind of a print, some kind of output. And this output will be actually used for as a prompt to the agent, to the language model. And I have this language model setting, not this one, here, right? So it's OpenAI models. You can see that GPT 4.1. And then it's produced this, second, if you do prompt engineering, you're familiar with this. You're a helpful assistant to help the user to operate the automated imaging lab. And then we also give some tools to the agent. I'm gonna hide it so it's easier. So we also have some tools to allow you to query these PubMed, for example. I'm gonna do this. So if we want to study the cell morphology changes, if we, after taking the cell out of the incubator, can you look for PubMed for some literature? PubMed, yes. So this will I just do a quick demo to show you how so it generate code immediately. It's query PubMed, and it's find some papers, right? And then trying to summarize for you. So you can use this a lot of tools. We connect to archive, maybe not by archive yet, but a lot of tools which enables the agent to help you generate the hypothesis or brainstorm ideas. And then in the meantime, yeah, okay, so it started to already open the lab. So this is our lab in Sweden. So it will start to load the plate. This is the incubator. This is the robotic arm, and this is the microscope. It's trying to reset the microscope now. So this video is not generated, yeah? It's real. Let's see. Finger crossed. So you can see there's something running there, right? There's a door open to deliver the plate outside this incubator. The robotic arm gonna take it and then put it to the microscope. So this is our microscope. Not as fancy as yours, but it's just a bright field or wide field microscope. And our aim is trying to demonstrate the capability before we get a bit more complicated to make a more robust system for live cell imaging. Okay, so now put on the microscope. So it's navigate to well D4. So it's a 96 well plate. Why it does that? Because we have some prompt, give some kind of instructions. So it started to get an image. Oh, yay. An image from Sweden. Okay, so the cells are dying, right? Because we tested many times, and then the cells cannot. Now you can do image analysis. I'm gonna show you. Could you please do a segmentation on this image? This is definitely not staged, huh? I never tested. Okay, so it's trying to read it. And the cool thing is that you can edit the code, yeah? You can also learn from the agent if you, or you can make changes to this. Okay, we have some problem. Yeah, it's missed this scikit image library, and then it's trying to install it, right? I'm gonna actually, yeah, it does, what is it? This is actually very bad. Could you try it better? Because this is a white bright field image. The background is a bit lighter, and the cells are actually a bit gray. Could you try to find the cells in a bit, a dark color? So the goal is that I'm not trying to show you that a perfect staged demo, but the idea is to show you that it's actually quick to allow you to navigate or work with the system. By no means is that it's more smarter than us, but it's definitely quicker. Yeah, it can make this code very quick, and then it can interact quickly with you. It can kind of iterate with you. Let's see if you are luck. Okay. Yeah, okay, I'm gonna stop here. But you get the idea, right? So you should appreciate the kind of the speed or how quick it is, right? Imagine you talk with a student or someone, ask, hey, could you please fix this or something? And this is much quicker. All right, so you have seen the demo, and this is it, my backup demo. I don't need anymore. And next I'm gonna show you another demo, which is a bit what I'm thinking, kind of the way, it's a better way for us to interact this kind of system. Okay, so let's proceed with the study.
Great. Let's navigate to a well and start imaging volume, to assess the U2OS cells.
So this is a voice interface. Yeah, you can see that it's a bit like ChatGPT voice, but it can interact with it quickly, so you don't even need to type.
Outside the incubator, the microscope is now auto-focused. Let's snap an image to begin our analysis. Here's the image of U2OS cells taken in the 561 nanometer channel. Let's proceed with analyzing this image.
Yes, go ahead.
Let's analyze the image to assess cell viability and morphology. We'll look for indicators of cell health and stress responses. Let's continue. We've identified 14 cells in the image. Their morphologies vary, suggesting different stress levels. Some cells may show altered shapes, indicating stress from being outside the incubator. Shall we continue with more images or further analysis?
Yes, maybe move to another well and, for example, A4. And then let's take another image to get more statistics.
Let's move to well A4 and take another image.
Yeah, go ahead and navigate to A4.
Here's the image of U2OS cells in well A4. Let's analyze this image to gather more.
My voice is definitely more excited than that. Was taken during night. Okay, sorry. So yeah, so I hope you see the future we are targeting, that we want to build this kind of agent-operated labs. So you have a lot of AI agent, like lab technician, and it's connected to a lot of databases, Microscopy, the AI agent, data analysis agent. And then the whole plan is trying to let the agent run the lab automatically from end to end. So it's a little bit goes in the same line as what Florian was talking about yesterday during this focus on trying to have this loop. Yeah? So this is also very kind of an active field. It's only possible with this AI agent. It's called AI scientist. In the end, we will have a lot of AI scientists running our labs, and then humans need to maybe slightly shift it a bit role to work with this agent, a bit like what I was saying, hey, go ahead, something like that. And then, you know, for our lab, we're trying to collect a lot of data in this combined with real-time analysis and trying to build this virtual cell model and going back, provide insights, and then instruct the next step. So in the future, we will try to close the loop and kind of have an end-to-end system operated by the lab, by the AI agent. All right, I think this is my last slide. I have been showing you different possibilities of video diffusion model. This is just for modeling the cell in a black box manner. It's not ideal, but this is what I think when you try to push performance, you need to sacrifice the kind of clarity, explainability, interpretability. And then I also showed you automated data acquisition with LLM agent. And then with this, hopefully you can see this kind of a automated scientific discovery, how it looks like in the future. I would like to thank my lab members, PhD student, master students, my postdocs, and also our collaborators, especially Manu, Emma from Stanford, and Hun Chuan from this Cephala, also Stanford, provide this microscope to us. And of course, the funding agency. Thanks for your attention.
Thank you very much. I'm a bit old school here, so I'm not using very much ChatGPT, or I mean a little bit, but not so much. And we always have a lot of discussion in the lab asking ourselves, well, shall the students use it? How do we approach the students? So I guess my question is a bit more ethical. So do you think this would be like a tool that the students, for example, and also the PhD students should really use instead of scratching their heads and, you know, like writing things and trying to solve the problem themselves? Or you think it's really something that could be useful for the next generation of scientists?
I think that's a great question. I'm also doing teaching. My answer is definitely, you know, all the student or all of us should, you know, actively adopt this tool. But, you know, we need to use it, you know, properly, responsibly, but also properly, meaning you need to learn this prompt engineering, you know, you need to know how it works. I think it's just make things much faster, right? Let's say consider this, right? Programming is something we created, right? To force you to think like a computer. In a way, it's a bit inhumane, right? Because you're trying to force to think like a computer. If there's something replace it, then you can do natural language, produce this code, and then you don't care anymore. But of course, you need a way to validate the result. But often also I talk with the student that you need to learn how to use it. Most importantly, you need to know how to validate the result, because it can easily fool you. It can produce hallucination, and you need to understand it very well, and you need to learn some technique to how to cross-check. I wouldn't worry about that because the biologists are used to work with the black box. Right? It's the kind of cells you don't understand, but you design control experiment to do. So the same kind of methodology would apply when you work with this kind of generative AI agent, for example. You need to, when you ask it to do something, you need to have a way to test it, validate it. So this is what my kind of takeaway so far.
Do you have some?
Thanks a lot. So yeah, very interesting. So I get the part about the automation of analysis and microscopy, and also the part about AI agents, but I don't quite get the first part where you said you want to create a virtual cell, and you want to create a tool that you gave an example, like you would give the first image of an actual video, and you give it a prompt, and you said you want the model to reproduce a video where the human eye could not make the difference. But what's the use for this? I'm sure there is something, but I don't quite get the interest.
Yeah, sorry about that. Imagine this: if we have this generative model, you can feed in a drug molecule, the equation, or kind of smile string or something, to the model, and this model gonna produce a movie which the cells are dying, or produce a model a kind of cancer cell is being killed, but a healthy cell is kept alive. So this could be easily used for some in silico drug screening, for example. And then you can, if you can expand this kind of database, you can do a lot of in silico cell experimentation. Now you can do a lot of, you don't need to go to the wet lab. You just do this in silico, and then you can understand. It's very reproducible. It's very kind of much easier compared to much faster, for example. Does that make sense?
Yeah, but so the underlying idea is that by training a model to produce realistic outputs, videos, it kind of learns the reality of the underlying physical worlds. Is there a reason to think this?
Yeah.
Okay.
Yeah, it's like a record what should happen. Can all predict the future. If you add a drug to the cell, what is going to happen without doing real experiment.
Okay, thanks.
Thank you for the great talks. It's very impressive. So I have a question on, so let's say I have a straightforward experiment I want to do. I have the image and I ask for pipelines that do detection, tracking of dots, diffusion analysis, and then an histogram. And that doesn't make sense. The result doesn't make sense. I do my check, and now I have thousands of lines to debug to know where the issue is. Mm-hmm. So how do you, and it's very useful, I don't want to go from scratch, so you provide pointers of where mistakes may happen, perplexity, or do you have any trick to solve the problem?
Yeah, so this is normally how we work with the agent. For the first question as well, I mean, it's actually, I think this kind of AI agent actually make writing software harder in a way because you kind of lose control, right? Mm-hmm. The code is you don't write it. And then what's worse is that for the younger generation, they don't need to write this kind of code, making these very stupid mistakes many times as we did anymore. And then this is actually making it super hard for them to learn. So when we work with this AI agent, cursor and things like that, my kind of immediate feeling is that if someone never do this coding, they will never be able to use this tool well. So it's a challenge for in general, I would say, that how can you still game this kind of high-level thinking logic without actually doing coding? I think it's a general issue of this Gen AI tool. But coming back to your question, I think, so you use all type of ways and you're trying to instruct. The easiest is that you give the arrow stack trace to the model, right? And many of these AI agents, they do it automatically. So if you use a good agent, then normally if you just need to define the criterion. What is the success criterion? When we make software with AI agent, we define this kind of a test first. It's a bit like what they call test-driven development. So before implementing the software, you write a test. Let's say you want to write a snap image function from the microscope. You define a test case that the return of this function should be an image, right? The image should look like this and that. So this is the test you give the model, and then you let the model explore all possibilities until it matches what you expected test. Then it presents you the output, the outcome. And then if since still goes wrong, then you need to, work with it. And we have been kind of stuck for many times. Sometimes the agent can never get out of it. It actually makes it harder. Then you have in trouble. Yeah.
Thank you.
Do we have some more questions?
This one.
Back there.
Hi, thank you. Very nice talk. I have a question on the zoo that you're creating to let people label. I think the problem is labeling data nowadays with the intelligent microscopes.
Yes.
So, I want to talk on the ethical side of it. If you go to human cells and you want to put them online, I know in astrophysics we had, like, you put images of galaxies, so we call it Astro Zoo, and you let the public classify them, and I think that helps. But when you're dealing with humans and cells and things, where do we stop in terms of ethics? And I don't know.
You mean the privacy and
yes.
Like that. So actually, this is not a public link. Yeah, there's a super long because it has encoding. It's the same as you upload to Google Doc and you share with your colleague with this private link or something, or it's the same as you upload. You have your email on Google, right? And you trusted Google. Sometimes, or sometimes you don't. But this is kind of a standard security measure we have. So you share a link, which is super long. No one can guess it. And then you share with your colleague. And then the image is only transferred from your computer to your colleague, right? The trusted one. So HiFi actually have a lot of effort trying to make this kind of privacy preserving possible. For example, there's a special technique called WebRTC. It's enabled like peer-to-peer connection. So your image will not go through the server running by us if you don't trust us, right? And then it will actually go directly to the ones you trust. But thanks for the question. Just maybe one last, some kind of ads. We are looking for PhDs and postdocs for this automated imaging system. So let me know if you're interested.
I might have one last question. Thank you for the nice demo of your automated AI imaging system. Can you maybe give us a quick oversight on how long it took to make the agent be able to steer the microscope and understand your commands? Was it very easy, or can I very easily implement it in my own lab?
So it's actually straightforward. I think normally you need to, first of all, you have some kind of Python functions. And the most important thing is you need to write documentation for those functions, for all the arguments. And you need to, and often the mistake coming from that you can assume a lot of knowledge because you're stuck in that developer mindset. So it's helpful to find someone else. Just to look at the text, can you figure out how to use those tools as an agent, right? So if you can pass that, you ask anyone, look at this, hey, I kind of understand how it works, then you are fine. Yeah, you define this documentation, the agent gonna pick it up, and then it can normally, figure it out. And the agent also quite fault tolerant. If you give some kind of wrong documentation, it will try first, and then it will hit this error, and then they're gonna fix it for you. Normally, yeah.